Good afternoon, my friends. It's good to see you again. You are an amazing audience. I was blown away by your questions on Friday morning, and it was exciting to just be part of that. And the amazing part is that you're all back for more of this stuff. So I know Gerald is very good, but so are you. Very briefly, I will introduce Gerald once again. He's the provost, the new provost at Hope College. And just a minute. Can't see anything without those. He came to Hope in 2015. His research interests primarily focus on the reciprocal interactions between viruses and the nervous system, which is, of course, what he's talking about. He has a bachelor's degree from Cornell, a PhD in neuroscience from the University of Pennsylvania, where he also did a postdoc, and focusing on the herpes simplex virus type 1, his famous HSV1. And we welcome Gerald to our second session of this course. Thank you.
He's the provost at Hope.
Thanks, everyone. It's great to see you all again. And for anyone that's new, it's great to see you for the first time, either in person or online through Zoom. I want to start by just giving a brief intro, particularly for those who were not able to join us Friday, and just kind of as a way to recap, stress some major points. Today's session is going to be more focused on the interaction between the viruses and the host immune system, as well as some different aspects of behavior. We'll talk a little bit about impact on insect behavior, as well as some newish data from my laboratory here at Hope. It's nice to actually be talking about this because now in my new role, I actually don't talk about viruses much day to day. I get asked a lot of other questions, but not about HSV1 behavior in immunology. Some of the slides that you'll see today actually come from a class that I pioneered here at Hope called Psychoneuroimmunology, or P&I for short. Based on what I found at the time, and it premiered in spring of 2018 at semester, it was about the second or third undergraduate course of that same type in the country. So I believe Northwestern had a course, I believe Yale had a course, and Hope College had a course at that time. And it's really building. You see this course type at the graduate level study. So it combines several different disciplines. So psycho, behavior, as well as disposition. Neuro, we're talking about the nervous systems, both central nervous system and peripheral nervous system. And then immunology. So really thinking about both innate adaptive immunity and how they could impact the brain at different aspects of our nervous system, as well as how that relates to overall organismal behavior. As you might remember, and again, those who were not able to join us last time, viruses are indeed obligate parasites. So they actually need us or any other host that has a cell to be able to replicate their viral genome. And again, that could either be DNA or that can be RNA. They are not able to metabolize. They are not able to send or make new genomes on their own. They indeed need to be in a cell. Either they produce their own replication machinery, or they take advantage of our replication machinery to replicate and divide themselves. Again, a schematic of HSV1 or herpes simplex virus. I'm just going to say HSV1 for the entirety. We see this capsid here. And again, these glycoproteins that are pointing outwards, and that's where they're going to interact with receptors on our own cells so the virus can gain entry and able to replicate itself in lots and lots of virions or virus particles, as we refer to them as virions. And one thing just to note is that HSV1 is a large coronavirus, i.e. looks like the sun, and it is a double-stranded DNA virus. And so that's just something to take into account. It doesn't have a high mutation rate compared to RNA viruses. And so it's pretty stable. compared to other viruses that particularly SARS-CoV-2 or COVID-19 that we've been aware of. So it doesn't have an error rate or mutation rate that high. When I first began to teach this class, the P&I class at Hope, I had one segment on viral immunity. And I want to say, why do we care about chronic virus infections? For most of them, they don't do too much in terms of human health load. So we talked about infections such as herpes simplex virus, cytomegalovirus, CMV, you may have referred to it. CMV infection, actually, if a mother has CMV, an active CMV infection that has reactivated, the chances of the offspring having some type of neural insult or behavioral insult actually increases. So there are statistical data showing that actually having an infection can actually propagate behavioral changes from mother to child. Varicella, also we discussed that. And these infections can form latent bouts, and that can last, again, the entire lifetime of the host. I wanted to break down the latency step for HSV1. I kind of alluded to that last time, but I want to make sure that we can see it in its kind of visual depiction. We're going to use the arrow here, so forgive me, I will be behind the podium just for a little bit. But here's kind of the establishment of the infection. So kind of out here in the blue, if you will, is some epithelial cell or mucosal cell that has become infected with the virus. Again, the virus is not going to stay latent in those cells. It's going to multiply and go and eventually find its way to some axon terminals. Now, these are axon terminals. These are places where typically neurotransmitters are released. They're also the places where the virus can actually be sucked up, if you will. And then we have what I referred to last time as retrograde transport. Mouse does not like me. The orange represents the direction of the virus going from axon terminal to eventually the soma, which contains the cell body and the nucleus. Now, we also see here a protein focused on HCF1. HCF1 is host cell factor 1. It's a protein, also it known to interact with, again, get my cursor here, one of the viral proteins, VP16. One interesting historical note, there's lots of abbreviations in HSV1 viral protein nomenclature. These abbreviations used to stand for things. Now they do not. So they're just really known, the protein's name is VP16, but before that it was viral protein 16. VP16 is a protein that is going to be reproduced in high amounts early in infection, and it can bind to the HCF1 factor protein, shown here in these black dots, allowing the virus again to go through the axon. These orange blocks, if you will, just depict the myelin sheets. So myelin is a substance, a fatty substance that helps insulate the axon so that nerve conduction can go really quickly. But we're going the opposite of nerve conduction, right? So nerve conduction is typically going to go from cell body to axon. We're going from axon to cell body, hence the name retrograde transport. In the establishment of HSV1 latency, one important step is that there are silenced HSV episomes, all right? So we can see here in the nucleus that the viral DNA gets into the nucleus. It's not binding directly to the chromosomes. It can at some point, but typically it stays with it, episomal. So the HSV1 DNA is going to go to the cell body and go into the nucleus. Upon reactivation, right? So if you were stressed, either emotionally, physically, or temperature-wise, the HSV1 DNA, these episomes are going to be reactivated, and then we see some of that HCF1 factor being produced along the way, and we start to produce with it these HSV virions shown here, which are then going to be released from the axonal terminals, and then you'll see the cold sore reactivate, or wherever the virus initially infected, you'll see those sores come out. But what happens during latency? Right? So during latency, we have episomal DNA here. But what we also see here, what I showed last time too, are these CD8 positive T cells, right? These satellite cells that surround the infected neuron. And as you might remember, these infected neurons are sensory neurons, okay? So they're outside of the central nervous system, they're in the peripheral nervous system, either in our trigeminal ganglia or in our dorsal root ganglia. You see, based on different amounts of data from multiple labs, that it's important to have some neurotrophic or trophin signaling. These are factors like brain-derived neurotrophic factor. They help keep neurons healthy. They keep neurons in high maintenance. One thing to remember is that neurons, unlike other cells, they don't replicate. There's one exception that we could talk about, but for the most, they do not replicate. So once you have a neuron, you will have that neuron for the rest of your life unless it dies, and then you will not have that neuron anymore. If there is some type of environmental stress shown here, that's going to kind of, it usually would signal a reactivation, but because of the presence of these CD8 positive T cells and their immune effectors, these cytokines that we mentioned earlier, interferon gamma, here we talk about granzyme B, which normally actually pokes holes in neurons. It doesn't do that when the cell is infected with HSV1. And so this allows for the virus to stay latent and only produce one small piece of its DNA. So that's essentially kind of the beginning of this interaction between the immune system and a viral infection, which you actually need to promote latency. And so this is a little emoji of me. So how I capture, as soon as like this goes against all what we know about cellular immunology. Granzyme B, like literally the name means to like zip it, like it literally pokes holes in cells. It's supposed to kill it. Interferon gamma, it's known to kill cells, including neurons. What is going on, right? How can we explain this? Well, one thing we can look at is that during chronic viral infections, CD8 positive T cells that are releasing these cytokines express high levels of programmed cell death protein 1, as well as interleukin 10. I abbreviated it as IL-10. Also another type of cytokine. Interleukin 10 is known to inhibit the activation of immune effector cells, okay? It is an anti-inflammatory cytokine. So perhaps there's some balance. You have interferon gamma, you have granzyme B. Now you have some interleukin 10, perhaps they are opposing one another. And maybe this is one method, one mechanism where that accounts for the lack of cell death, perhaps, right? PD1, programmed cell death protein 1, promotes apoptosis. This is regulated cell death. Okay, this is the cell decides that there's something awry, and the cell has a trigger of genomic and proteomic cascades that's going to cause for its own death or its own suicide. And so PD1 promotes this cell death in antigen-specific T cells while reducing apoptosis in regulatory T cells, right? So it suppresses inflammation as well. So these CD8 positive T cells, they get to stick around, thanks PD10, and you're suppressing overall inflammation state, even though you have a chronic infection. And these point to possible some putative mechanisms that may be able to reconcile this information that we know, granzyme B, interferon gamma. They're supposed to kill cells, but we don't see that. And in fact, when you remove them, you actually get a reactivation. Another one that I think maybe a more likely target is this phenomenon of T cell exhaustion, right? So over a chronic infection, you have days, months, years, lifetime of a host or hosts that are infected. What do you think is actually happened to the T cells? They're activated. They are, they know something's going on with the infected cell. They surround these infected neurons. But T cell exhaustion posits that you eventually will get progressive loss of T cell function. There's a little bit of interferon gamma release, but not a lot. And over time, perhaps not too much, but the CD8 positive T cells are still there. perhaps leading to the removal of responding cells. So much of what we know about HSV1 mediated immunology is acute infection and beginning of latent infection. We don't know so much about the lifetime of the host. So perhaps if we were to infect the mouse, as we did in the earlier studies, not only look at 30 days or 45 days post-infection after initial beginning of latency, what if we look at 200 days post-infection? What if we look at 300 days post-infection? Mice typically live only about 2 years, so we can't look up much longer than that. But perhaps if we did a longitudinal analysis of the plasma as well as the trigeminal ganglia, what do we think we'll still see? Will we still see CD8 positive T cells surrounding these infected neurons? We don't know. We don't have that information yet. Lastly, we have some information from one group showing that these T cells are still present in trigeminal ganglia, but they become functionally inept. So again, they're not doing what they normally do. They're not releasing these cytokines at the appropriate levels that you might have thought they would. Here's a graph. I'm sorry, but it's a little bit off the screen here. But on the y-axis, and I'll do this for our viewers online, we have number of virus-specific CD8 positive T cells on the y-axis. So that's on the y-axis in both A, B, and C. And in yellow, we have a cartoon of the amount of viral load. So you see here in acute infection, there's a rapid increase, and then acute infection, that viral load goes down, right? You get infected and it goes away. That would not be HSV1, by the way, right? That would be, say, influenza. You get an infection, you clear it, it goes away. Then you have in B, protracted. In the black, blue, and green and red lines are representing levels of CD8 positive T cells that are specific to virus, interferon gamma, TNF alpha that we'll talk about in a little bit, which stands for tumor necrosis factor alpha, and then interleukin 2, black, blue, green, and red respectively correspond to those levels. In the protracted level, you see a rapid increase in viral load, and then this steady decline in viral load over time after infection, which is on the x-axis, okay? So eventually you do clear the virus, but it takes a while. And then lastly, we have C, which would be the case of HSV1, which is this chronically infected state. There's a rise, and then you actually have this steady presence of viral load, viral protein, or viral DNA present. And again, you have this increase of T cells over time, not rapidly, but slowly going down. And its activities functioned by the levels of the cytokines also going down as well, representing this T cell exhaustion. I'm here, I'm ready, I've responded to the infection. Oh, you're still infected. I don't have any more to give, right? It's like they came, trying to find a good analogy to it that, let's see here. It's almost like the Amazon delivery person, comes with the truck, delivers, but you just keep ordering more and more, and they just can't keep up because maybe their truck breaks down, I'm not sure. Or maybe milkman. I know people don't have milkman or people anymore. You just run the supply out, right? There's no more milk. There's no more granzyme B to give. And so therefore the cells are there. They're ready to do their job, but they can't keep up with the persistent presence of the virus. So why do we care about this? Well, they can have some really long-lasting effects, these chronic viruses. Some viruses are known to increase plasma levels of tumor necrosis factor alpha, TNF alpha. This cytokine has been shown to increase the permeability of the blood-brain barrier. Well, why is that a problem? Well, our brain is a very immune-sensitive organ, right? There are really no immune cells of the brain. There are some microglia, but they're not going to rapidly destroy an infection, if you will. They will destroy neurons that are infected. And so you begin to get neuronal loss, or you get to have gliosis, which is just a rapid activation of the glia, and they form these clusters, which are cytotoxic to the neurons. And so again, the end result is neuronal loss. So as we make the blood-brain barrier more permeable, we begin to get a decline in actually neuronal activity, which will be related to cognitive function, motor function, just depending on where in the cortex or where in the brain you have that neuronal loss. We also see that in some very young individuals, they are not able to mount an adequate type 1 interferon response to an infection, okay? And then we stimulate these dendritic cells, also producing interleukin-10, and again, just causing multiple types of disarray in the nervous system. One thing we can look at is actually plasma levels of patients who have major depressive disorder. So on the y-axis, and I'll go back to the laptop here, we have concentration of tumor necrosis factor alpha. This was taken in patients that have been diagnosed with major depressive disorder, or their age match control. So they did not have major depressive disorder. And so on the x-axis in these blue circles here, we have the average and range of plasma levels of TNF-alpha in those controls, healthy controls. We have levels of TNF-alpha in those individuals. There are 58 in this study that have been diagnosed with major depressive disorder without any treatment. So this is baseline. And you note that it's statistically significantly greater, the levels of TNF-alpha compared to the control, right? So they excluded people who had any type of addiction, any type of history of abuse. There were other exclusion criteria as well. So they wanted to make sure that this was really the impact of major depressive disorder and not other covariants. What is more telling, at least to me, is the data that follows. In the orange and the green dots, this is post visits after they've started treatment. Most of these patients receive some type of SSRI, selective serotonin reuptake inhibitor, or some type of SNRI. So instead of increasing amounts of serotonin only, they also increase levels of norepinephrine, both being shown to ameliorate the impacts of major depressive disorder. So we see a behavioral change as well, or sometimes not so much at the 2 weeks, but definitely at the 12 weeks, some behavioral changes were noted. But even before behavioral changes were noted, what do we see? A decline in the TNF-alpha levels. Again, taken from plasma, not from the brain. These are circulating cytokines that are responding to infections or being ready to respond to infection. And we see already a difference between healthy control and those patients with MDD, major depressive disorder, thus suggesting a link, right, between the plasma that circulates all of us and the cytokines that are in there in terms of our disposition, in terms of our predilection for anxiety, depression, etc. So the more we understand about our immune responses to an infection, I believe it's the more we're going to better understand some of these mental disorders and mood disorders as well, because we also see similar things with anxiety as well as MDD. I got the question the other day about how did we really know that these animals that we were studying were actually in latency, right? And I wanted to break down kind of our thought process, because what we did is that we looked at the trigeminal ganglia of the animals after we had performed the behavioral analysis on them to see what their levels were of two particular genes, one being the thymidine kinase, but also the major one being LAT, latency associated transcript. And so that's what this LAT shows here. So in the epithelial cell, we see VP16 and HCF localized to the nucleus, excuse me, that was in the earlier figure. And these immediate early genes, that's what the IE stands for, are expressed. That allows for the virus to replicate rapidly and at high levels. And so VP16 and ICP0, and again, it stands for nothing now, but it used to stand for infected cell protein 0, which are very early genes. They help to reduce heterochromatin formation. And then the HSV genome associates with euchromatin, so true chromatin, which basically just means the DNA is flat and ready to be expressed at high levels. In neuronal cell, we see VP16 and HCF in the cytoplasm, which will be all throughout here. So kind of the liquidy matrix of the cell. Immediate early genes are expressed, LAT is expressed, and during latency, we get only LAT being expressed, promoting heterochromatin formation. And a former postdoc and good friend of mine actually showed this quite well in the work that he was doing as I was at University of Pennsylvania, showing that we actually had virus that actually had LAT deleted, and you didn't see this heterochromatin at the high levels, at least. And so we know that during latency, the viral genome is able to associate with heterochromatin, so modified DNA and its proteins, basically modulating our own gene expression. This is in one particular note because we can utilize this because we have a number of herpes viruses that have been modified to target cancer cells, so those cells that are oncolytic. And so there's a lot of herpes virology work that is utilizing the replication cycle of herpes virus to actually target and kill cancer cells. A project that I was working on, we actually used a viral vector. So we had a piece of the virus that we took out and we put in there a gene that was actually missing from a mutant mouse. In this case, it was a mutant mouse that was replicating the effects of one of the lysosomal storage enzyme diseases. So these lysosomal storage diseases can have severe effects on cognition, but mostly also skeletal growth. And it's has pretty profound effects on development of young children. And so we were able to put in this missing enzyme into mice that didn't have it through the herpes virus, put it directly into the brain of these mice, and actually showed that we were able to impact their behavior for the good, so increased memory, working memory, short-term memory, when we did this using this herpes-based viral vector. I want to take some time to shift how we are thinking about the relationship of, or the etiology of dementia, particularly Alzheimer's disease. Alzheimer's disease was really the impetus for me getting excited, interested in biology. It was my grandmother's experience with this that led me to wanting to learn more and more about the brain. And here I am, looking at how HSV1 impacts potentially some causes of Alzheimer's disease. What we do know is that there has been shown, I talked about this the other day, that the DNA of HSV1 has been shown at high levels in those who succumb to Alzheimer's disease compared to healthy age matched control, healthy age and gender matched controls. We also see that amyloid beta plaques, these are the plaques that have been long associated with all types of dementia. But moreover, we also see these neurofibrillary tangles, or NFTs, if you may have heard of them. They have been more specific to Alzheimer's disease compared to others' dementia. And what we see is that actually these plaques and tangles actually have bind to HSV1 proteins. And so it's an interesting kind of correlation about why are these plaques and these tangles binding to HSV1 proteins. Here's some micrographs from this is from the Wozniak lab in the UK, where they showed, I believe these were Vero cells, or just kidney cells. But what we see, the blue dye just represents the cell bodies. So they're representing or the nuclei. They're representing all the cells present. And the brownish tint is showing the presence of amyloid beta. And so this is from brain specimens with dementia, and you see really high levels of amyloid beta, as well as really high levels of phosphorylated tau. And the phosphorylated tau is the form of the protein that has been found in the neurofibrillary tangles, or NFTs. What we see is that HSV1 latency prompts an increase in TNF alpha as well as interferon gamma levels. So we've kind of alluded to this earlier, but we see it in intact rodents as well. We also see that interferon gamma increases inhibition of neurons in the prefrontal cortex, really important for working memory. And so there's kind of this two-step connection between HSV1 and memory, but now we actually have some more detailed analysis and results. What I have on the screen here is beginnings of some research from my lab with undergraduates here at Hope, focused on phosphorylated tau. We have the neuron here, again, axon down here, cell body right here. And in the axon in particular, we have these structures called microtubules. And they are made up of, there are multiple subunits of this protein called tau, TAU. And tau is needed to keep these microtubules together, but they can also, the tau can be phosphorylated, as we see here in this red block. And then the phosphorylated isoform is shown here, the phosphate group, is shown here, this P, it's a little gray, so you can't see it, those are Ps there. If we get hypophosphorylation of tau or too much phosphorylation, what we start to see are these filaments, which can lead to these tangles, these NFTs. And it's these NFTs that are highly neurotoxic. But again, it begins with the hypophosphorylation of tau. What we found here, and these are kidney cells, but we also have some data from rat hippocampal neurons, is that cells that were uninfected, we see a little bit of tau because tau is indicated here in red. And the blue is a staining that binds to DNA. So it's showing all the cells in this Petri dish. And then cells that were infected with HSV1. So notice you'll see less cells because the virus does kill these cells. These are not neurons. But you start to see some clustering of really bright red in these places here. And so when we actually quantified this or did semi-quantitative analysis, we looked at the density of phosphorylated tau, okay? So these are density of phosphorylated positive cells or labeled cells. So the number of cells that were labeled with tau divided by the total number of cells in a particular area. And what we see that with uninfected cells, very little levels of phosphorylated tau. I'll back up and say that this work was funded by the Department of Defense, particularly the Army Research Office. And so they were interested in what might happen if a soldier went to war and was exposed to some type of traumatic brain injury or a blast, a sonic blast. And so we mimicked this in vitro in the Petri dish by adding sonic waves to that dish. And then we also did physical disruption in some other studies. And so we also damaged these cells as well. Physical damage, either by sonication or by physical damage, that was like with a razor, increased levels of phosphorylated tau, which is to be expected because with damage, and particularly these viral cells, they can replicate. And so you have combining of the microtubules, which might lead to hyperphosphorylation. We have infected, no damage, both higher than uninfected, no damage, which we also expect because, again, the virus is impacting the cellular structure. And then when we have infected and damaged, okay, so this would be the soldier who already has HSV1 before going on their tour, and then also comes into contact with some type of sonic blast, some type of mild, even moderate traumatic brain injury. And you see additional higher levels. So they have this synergistic effect with one another of increased levels of hyperphosphorylated tau. We also see this just in the staining amounts of tau as well, or phosphorylated tau. And so this is some beginning data in the lab where we were going on an avenue to see if we could determine how these two different events work together, either virus and brain injury. I want to also talk a little bit about insects, if I can as well. So in the class with psychoneuroimmunology, I like to expose the students to different types of immunity systems, right? So we talked a lot about mammals, but also we did a little bit of work in arthropods, mainly because they're so important even for how we come in contact with viruses as well, right? West Nile virus, et cetera. So we talk about viral vectors. Our natural viral vectors are these insects that potentially bite us and then can spread disease. One thing to note when we do this, we talk about vectorial capacity. So these are about the insects themselves and their population's capacity to transmit an infectious agent to a new susceptible population. And there are multiple factors that can impact this. So how dense the insects are, how likely they are to feed, when are they feeding, how long do they live, their competence for actually holding the virus. So will the virus actually kill the insect, or are they able to keep it in their systems and not have their livelihood impacted? And vector competence, right? So the ability for the vector to become infected with and transmit a virus, follow an infectious bloody meal. So the mosquito bites you and is able to get some of that blood and maybe transmit it to another person. And how likely will they actually be able to transmit from person to person or animal to animal? Multiple places in the cycle here. So we have, say, vector, vertebrate hosts, viruses coming in, different stages of the vector, right? So different things that can impact how likely this vector is able to transmit a virus to the host and to multiple hosts. But these viruses not only make the host sick, they also alter the insect's behavior. And I thought this is really fascinating when we looked at some of these findings. So mosquitoes infected with La Crosse encephalitis virus actually probe more during feeding attempts. So the proboscis, right? Why do you think that would be maybe evolutionary advantage, right? To go in maybe at a higher frequency to actually transmit more virus to the host or get more blood so you can transmit to another host. Barley yellow dwarf virus makes aphids prefer non-infected wheat plants. So they're able to sense those that haven't already been infected. And so they tend to have an inclination to go there and infect those plants. Again, impacting sensory and motor systems of these insects. Dengue virus increases locomotor activity of Aedes aegypti females. So again, making them more mobile, perhaps increasing their activity. And these are all things that we can begin to build upon, that we see viruses not only impacting mammalian systems, but also arthropods that many don't have a true brain, but do have some rudimentary types of nervous systems. And so with that, I will kind of stop and we can take questions, but the overall goal really was to help make these links between both, number one, immunology and virology, as well as showing different examples of how multiple viruses can impact both mammalian behavior as well as insect behavior as well. So with that, we can have some Q&A if we have time for that. I think there's a question.
It's very interesting about this barley yellow dwarf virus with aphids.
Mhm.
And then they like to go to the wheat plants to feed on. So they're actually crossing from the preferred barley to wheat. They're
Yes.
kind of like changing the diet.
Mhm.
Wow,
Definitely.
that's very interesting. Yes. Thank you.
Yeah.
We have a question from online. If someone on a medication that blocks IL-1, can that increase the viral load or affect the infection in another way?
That's a good question. Potentially, yes, because then you're changing the composition of all the different chemical milieu that would either be pro or anti-inflammation. And so that could prompt changes in how fast certain structures are produced. One structure that has gotten a lot of talk in the past few years is the inflammasome. So it's a cellular structure that once formed causes a whole chain of chemical events that could lead to the destruction of the cell due to some type of infection or presence of cancer. So inflammasomes are also produced in the presence of cancer as well. So if plasma levels of IL-1 are indeed changed, that could change sensitivity or the threshold to how quickly an inflammasome is formed.
Has anyone, I mean, I think a lot of these people are still living, but people with Havana syndrome, has anybody looked to see if they have an increase in hyperphosphorylation in the brain? And is there a way to image that without a brain biopsy? I don't know.
Right. I don't know if anyone's looked at that. That's a really great question. There are increasingly more ways of looking in vivo, in the intact living organism at these levels. I'm familiar with one for amyloid beta. I'd have to double check to see if one has, imaging system has been for phosphorylated tau. Yeah, great question.
Yeah, I have two questions. One is if the virus can produce all these neurofibrillary tangles, then I think you're talking about the central nervous system only. And so not that many people get the central nervous system herpes. So just because you have herpes doesn't mean that you will have this problem. Am I correct in that?
So it depends on, I would say there's a missing gap of knowledge that we have. So we know that the viral DNA is in the central nervous system. There's no sign, to my knowledge, that there's intact active virions or intact virus that is replicating in the central nervous system. Otherwise, you would see encephalitis. So we don't see that. So the question is, what components of the virus are getting to the central nervous system? How is it getting there? Because we do see these viral binding proteins, and we see them. There's another paper I was looking at years ago. They were doing this, they were co-immunoprecipitating. So they were pulling down proteins from the brains of those that had succumbed to Alzheimer's disease. And there were viral binding proteins, and stuck to them were HSV1 DNA. And so that's in the central nervous system. How it got there, why it's there, is the big question.
So anyone who has had HSV1 potentially has components of the virus in their central nervous system.
Potentially. We don't know how widespread it is, because these were very progressive cases of Alzheimer's disease. So maybe it's a certain severity that is associated with that. I mean, this is related, but a bit tangential. So we do know that circulating DNA can get to the central nervous system. And we know this is from evidence from mice that if a mother is pregnant with sons, we see male-specific DNA in the mother's brain. right? You don't see it if the mother didn't have any sons. And so that means that there is some circulating DNA is able to get through, number one, the placenta, but also the blood-brain barrier. So perhaps it's these DNA segments, right, that are able to circulate and get to the brain, whereas they would not be able to get into the brain if it was the fully formed variant with the proteins and the glycoproteins, et cetera. So it may just be the DNA getting to the brain, which would render itself directly not harmful, but if it's causing for increase of NFTs, then it would be.
And the other question I had was about the T cell exhaustion. Is that why doesn't the T cell kill the neuron right off in the initial?
Yeah,
In the initial thing,
yeah,
which would be disastrous,
right.
for the organism.
Yes.
So I'm thinking there must be some sort of signaling that this is a neuron that you shouldn't destroy, and so they just sort of hang out and don't actually go in for the full kill.
Here's my working hypothesis is that I believe some portion of viral DNA, I'm a fan of LAT because I've studied it for so long. So I would say that some part of LAT, latency associated transcript, which we know actually can regulate our own DNA because it produces what we call microRNAs, right? Which are just short stretches of RNA that are known to be regulatory in function, right? They don't encode proteins, but they actually regulate the expression of DNA. And so I would posit that it is LAT through its microRNAs that actually is able to regulate the expression of receptors for interferon gamma, right? You can have a lot of interferon gamma, but if you don't have a lot of receptors, it's not going to do too much, right? Or maybe it changes the receptors so that there is an increased threshold for activation. So those are two of my working hypotheses.
So, I mean, part of the virus, even though it's not living, there's got to be some, I don't want to, it wants to survive, right? I mean, it wants to be able to replicate again. And that's why we're seeing kind of these mutations in the coronavirus. Because if it triggered everything to kill it, then that's the end of it. And I guess there must be, I don't know, does that make sense that it has some desire for living on, so to speak, even though it doesn't live? I don't know. It just seems a lot of viruses mutate. And that's why I raised the question, because they just mutate. And we have to keep changing our approach to treating them. And they're always eluding us. So it suggests some purpose to that. You probably like viruses. So.
I do like viruses. So, right. So there indeed is this association with highly virulent viruses, right? Viruses that make people really sick and or could kill them. And their associated transmissibility, right? So again, if it's really easy to catch a virus, and if it also is going to kill all of us, it's bad for us. It's also bad for the virus, right? Viruses don't have brains, so it makes it tricky. And so it is always thought about of these random changes that get made, which one's going to kind of catch hold, right? And it really is just, if we can keep it at bay. So as we were going through the beginning stages, right, of the pandemic, I was reminding people, okay, each person are millions of replication, maybe I would say even billions of replication events for a virus. And if you look at the error rate, right, it's just, these variants were predictable. We didn't know exactly what it was going to look like, but we knew we were going to get variants that we were not going to be able to respond to, because the whole world was having billions of replication events in one person, and it spread so quickly. So I think the way I like to think about it is, if we can decrease replication, then it gives us a lot more time to be able to prepare for these predictable variants. You know, it's kind of like, you know, emergencies are going to happen. You don't know when, you don't know how, but can we have a response team, if you will, that is proactive. And so I think that's how I like to think about them. We can do microevolution studies in the lab because we can replicate viruses over and over again in cells, and we can kind of begin to understand maybe which mutations will happen faster than others. And that's based on the structure of the genome. And I should say, let me back up. Not only the sequence of the genome, but also the structure. How's it folded, right? So herpes likes to actually be modified and wrap around histones. It loves to kind of pretend to be us. Kind of makes sense because it's double-stranded DNA. Guess what's in our cells? Double-stranded DNA, right? So it loves being wrapped around. It loves these different modifications. And so it also likes, apparently it likes to be silent, but it's not completely quiet, right? It is replicating to some degree, just not the whole genome. And so perhaps that doesn't trigger, and because it's a double-stranded DNA virus with low mutation rate, maybe that doesn't trigger anything that would be too impactful for us because it is getting a signal, if you will, that I'm still doing work if we were to anthropomorphize the virus.
That kind of that sort of makes.
Yeah.
that makes the explanation.
Yeah.
And then you really tweaked my interest with the last one. So I saw a journal article. It was actually published on August 2nd in the Journal of Alzheimer's Medicine. And I think that one of the co-authors, you probably know her, and I'm not going to be able to say her name, Ruth Itziki at Oxford. So they were studying varicella, which is, by the way, the virus that causes shingles. So varicella, I mean, not varicella, that'd be a good name for it too. But anyway, it causes shingles. And they were doing this in mice neuroembryonic cells, a study. But at any rate, they found when shingles virus was activated, the varicella virus was activated, that it changed neuronal permeability. So perhaps then it could enter the brain. But they did mention in their studies that people that have had HSV1 who get shingles are at a much higher incidence of having these type of neurofibrillary tangles in their brain. So take home message for me was be sure to get your Shingrix vaccination. And by the way, if you didn't know, the old vaccination for shingles was not very effective, and you are eligible to get the Shingrix. You can have both, in other words. And so you might want to consider that because the Shingrix is, I don't know, something like 90% effective. It's 2 shots, but it's worth getting, especially if you don't want Alzheimer's. One more.
That raises the question, how many years old is the new shingles vaccine? That's
something like that.
so if I had a single shot before 12 years ago, I probably should consider the new one. Thank you.
This was probably covered in the first session, which I missed. I'm sorry that I did, but the herpes simplex virus one, how do you know if you have had that? And if it can be latent and you don't remember that you don't know of an instance you had it, is there a test that can show that if it's latent?
There's a test to show that you've been exposed. So it'd be an antibody titer test, and they could see if you actually have HSV1 antibodies. I phrase it that way because you could have been exposed and not had enough viral load to actually mount a full latent infection, or you could have a latent infection, but it just be a time period where there's very low levels of antibody present. So these antibody tests are known to not be quite sensitive. And so I just give that disclaimer that it's tricky from a a scientist's point of view, the best test would be for me to get in there and, and take some of your dorsal root or your trigeminal ganglia out. But we're not going to do that. We're not going to do that. But I would just say there's an antibody test if you are curious, and just know that caveat with it.
A cold sore, are there other, yeah. Yeah, cold sore. What are some other signs?
Right. Any
Okay.
blistering or itching. So again, you could have herpes anywhere where you have epithelial cells. So you can get the palm of your hand. I've seen it
Okay.
on the bottom of your foot. You can get it in your eye. It can cause corneal scarring. So you see that as well.
Okay, but it would be serious enough, you'd probably go to a doctor about it. It couldn't be so mild that you don't even, seek.
Some people
Help.
get a little itch and they
Oh.
never get like a robust damage of epithelial cells. And so that
Okay.
little itch could be the sign.
Oh,
And
okay,
yeah.
thank you.
Just you going back to your barley dwarf virus,
oh,
because it's kind of an easy one to talk about. You're saying that it changes the aphid's preference for food. Do you know if that's DNA related, if they're snipping and inserting something into the DNA chain of the aphid that makes it averse to the affected virus, the affected wheat? Or how does it affect the aphid that makes that change?
yeah, I don't remember. When you said the DNA change, that it sounded like that's what it was doing, but I'd have to go back and double check.
So that has many implications as to what else is going on.
Right.
Look at the lacrosse encephalitis. It's the same kind of thing that
Mhm.
could be making DNA changes, snipping and inserting small bits of information that make it change its viability or its activity level.
Yes, because if we look at arthropods' immune systems, they mostly are innate. Either they can clear it or not. And so it sounds like there would probably be related to some type of DNA alteration, either in sequence or modification of it. Yes. Fascinating, right?
I'd like to ask you just a general question.
Okay.
Have you read anything about the use of AI in some of the analysis of these? I'm thinking of the images you had up with the tau
Mhm.
and some of these other things. Maybe everybody read yesterday about the woman at MIT who's doing the research on mammograms. And the analysis is not scientific. It is strictly AI.
I haven't read anything specific to this and artificial intelligence. I would say that how we as laboratory scientists are measuring these different changes, more and more of it is becoming reliant on AI. How we analyze animal behavior. If there's open source software, Deep Lab Cut, it's free to everyone, and you can go in, you put your your animal of choice in video, and you can kind of tell the software to learn how to calculate, quantify different behavioral patterns. So I would imagine that we could also look at this in terms of, where those variants are being discovered and at what rate, and can we be, predictive validity? Can we increase that in terms of what mutations should we be expecting? And which ones may cause alarm versus those that may go silent?
Well, I will thank you, Gerald, for a
Yeah.
wonderful class.
Okay.
And
Thank you all. It's great to talk about viruses more. So, yeah. Thank you.
thank you.